Hello everyone and welcome to my Endocrine and Metabolic Diseases course. So first I will present the course contents. So we have 12 lectures of endocrinology, followed by 4 lectures of endocrine and metabolic diseases pathology, 4 lectures of endocrine pharmacology, and 4 lectures of clinical nutrition. So today is the first lecture of endocrinology, and in this lecture we're going to be seeing the fundamental principles of endocrinology. And stuff that you need to know to understand endocrinology, and we will also be doing a recap on physiology of the endocrine system and the things that you actually saw in your previous years of medical school, but things that you might forget. You might have forgotten. So the first thing we're gonna start with is what is endocrinology, and basically endocrinology. First, let's tackle what is it. So we have endo and crine. And endo means inside, and crine means secretion. So inside, secrete inside. And what are we secreting? We're secreting a hormone. So we're secreting a hormone inside the blood vessel. And what's the purpose of this? The purpose of this is to carry a message somewhere in the body. And we do this by using the blood vessel. So we can carry a message from a part of the body that is very far away from the target organ. That is the whole idea of endocrinology, and the endocrine system does this with hormones. So the endocrinology is basically a branch of internal medicine and a clinical science that deals with the physiology and diseases of the endocrine system and their secretions, which are the hormones, and their treatments, so the treatments of the diseases. So here we see a cell signaling modalities, and basically endocrine signaling is one of these cell signaling modalities, as I said before. So how do cells communicate with each other? A cell can communicate with itself with autocrine mechanism. So basically here the cell secretes something and targets itself. And a great example of this is basically the alpha-2 receptor. So whenever in the presynaptic neuron, whenever, for example, you're secreting a norepinephrine, if there's too much, the norepinephrine will bind to the alpha-2 receptor, and this will create a negative feedback mechanism. So that's an example to autocrine signaling. But also a cell, for example, the interferon signaling is a paracrine signaling, where the immune system cells will signal each other, but these cells need to be nearby. But endocrine signaling is the one that allows you to use the blood system, the vasculature, to access any part of the body. So as we said before, basically you can have one organ that is in a completely different location, and by using the blood you can reach the target, you can send a message. So basically the endocrine system is a system that allows to send messages throughout the body. And endocrinology will deal with this. And the fundamental pillars of endocrinology is this. So you need to keep this in mind. It's very, very important. So you have a signal, and this is basically how we control secretions. So how we control secretion of hormones. So you have a signal, and the signal can be anything, right? So it could be stress. It could be your circadian system, which we're going to talk about later. It could be a plasma volume. So your plasma volume might be depleting. You might have a hemorrhage, and this will create a signal. It could be anything, basically. So what's going to happen is that this signal is going to go and alert the endocrine cell, which is the cell that will secrete the hormone. Now the cell is secreting the hormone, which has a target, and then this target is going to then this hormone is going to come to the target, and it's going to tell the target, hey, you have to do something about this. And then there's going to be a biological effect. But this whole system has a negative feedback mechanism. So we say feedback here, but it's negative feedback, Because negative feedback protects you from situations where you're overdoing something, So let's say your blood pressure is low, You activate, for example, the renin-angiotensin system, And the idea here is that you want to counteract that low blood pressure. But if your blood pressure is too high, that is also something that is bad. So there's always negative feedback mechanisms associated to this system. So this is the main idea. Now, an example. So I'm going to give you an example where we have the hypothalamus pituitary axis, which we're going to see later what that is. But if you remember, the hypothalamus is going to basically secrete releasing hormones to the pituitary, and pituitary is going to release then pituitary hormones that go to the target cells. And then the target cells could be anything, okay? Because the pituitary gland is the master regulator of the endocrine system, and other endocrine organs will depend on the pituitary gland, okay? So you have your thyroid, your adrenal glands, your testes, your ovaries. But we also have some organs like parathyroid and pancreas that are independent from the pituitary. We're going to see all of this later, okay? But yeah, basically, this is, again, there's a negative feedback here, there's a negative feedback here, and there's also a negative feedback here. So this whole system is under negative feedback control, and this is very, very important. So here are all the endocrine organs. We're going to see them later, but obviously we have the hypothalamus pituitary axis. Very, very important. We have the thyroid, the parathyroid, the adrenal. We also have kidney because we have renin and erythropoietin, which will stimulate the erythropoiesis. This is for renin-angiotensin system. Skin is also endocrine because it is involved in vitamin D, and vitamin D, when it gets activated, it becomes calcitrol, which is also a hormone. So you have to think about skin. Your adipose tissue also secretes leptin, adiponectin, resistin. Your ovaries, your GI glands. We're going to see all of these later, but this is just a nice recap table. So we did see the secretion modalities, right? So basically we have the endocrine secretion. This is the whole idea. And basically we also need to classify hormones and understand what hormones are. So basically a hormone is a messenger, as we said, and we have to classify them. We can classify them upon their chemical structure, upon their site of production, or upon their biological activity. Now, chemical structure is very, very important. So chemical structure is going to matter a lot. So the chemical structure they could be peptides, they could be steroids, they could be amino acid derivatives, or they could be polyunsaturated fatty acid derivatives, okay? So here we see peptide hormones, which are basically coming from protein synthesis. Steroids are derivatives of cholesterol, and amino acids are going to be coming from amino acid, just one amino acid, okay? So you might be saying to yourself, what's the difference between amino acid derivative hormone and a peptide hormone? Isn't peptide amino acid? Yes, peptide is an amino acid, but the idea here is that peptide hormone is a peptide. So it's made up of multiple amino acids, whereas here we have just one single amino acid, okay? So we're going to talk about them, all of these. So first we're going to start with peptide hormones. And peptide hormones are produced as inactive hormone, and then they're cleaved into an active form and also other fragments. So this cleavage is a multi-step revision system that also allows the storage of hormones in vesicles. So this system is very intricate and very elaborate, but it's very, very useful. And why is it useful? Again, it is a multi-step revision system that also allows storage. And all releasing hormones from the hypothalamus, all the stimulating hormones from the pituitary, and their targets are peptide hormones. So whenever you're dealing with the hypothalamic pituitary axis, you need to think that, okay, they're peptides. Okay, this is a very easy way to memorize them. And in the future, in the next lecture, we'll be talking about the hypothalamic pituitary axis. So we're going to be memorizing them in detail. But for this lecture, I'm passing the slide. But this is a really nice way to memorize them. And once you know these hormones, you can just say they're peptides. But there are also some other targets. These are also peptides. You don't need to remember that they're all peptides. Like if you want, sure, go ahead. I mean, they're not that hard to memorize, but you don't need to, okay? But all of these are also peptide hormones as well. So yeah, that is also another thing. And basically oxytocin and ADH, antidiuretic hormone, is also from the hypothalamus. So yeah, that's also easy to remember as well. So you can think about all the hypothalamus things as peptide, but the others you don't need to memorize them all, okay? Steroid hormones, the second class. So these are derived from a cholesterol, and cholesterol is a lipid. So you can either obtain the lipids from your diet, where it's very from your eggs and your meat, right? Or you can get it from your liver. Your liver makes it. So physiology of cholesterol, very simply, is that it could be structural or functional, okay? So basically, it exists in the cell membrane as a structural lipid, and it gives it integrity. So that's its first structural compound. But also, the physiological functional aspect of it is that it's a building block to vital compounds. And this is very, very essential, okay? So cholesterol is used to make a bunch of stuff. And the most important stuff is you make hormones, okay? So steroid hormones need several steps of enzymatic reaction to develop from cholesterol. So we say that this is steroidogenesis. Okay, so you have cholesterol and you make steroid from it. So you have cholesterol, you make testosterone. Okay, and the idea here is that we have multiple steps of reactions, enzymatic reaction, just like the peptide hormones. We had multiple steps of reactions. Here we also do have multiple steps of reactions. So the idea here is that the clinical correlate here is that we have intermediate steps that exist. Okay, so we have some intermediate steps that you can see. So this means that we have production of precursor hormones with less known biological activity. Okay, so this is very, very important. And so we're going to talk about why it's important here in this slide. I'm going to talk and then I'm going to go back. So basically, there is the clinical implication where, for example, again, we have cholesterol, we're making aldosterone right here. So let's say there's an impedance in aldosterone production, okay? So here, aldosterone, there's an impedance. But what's going to happen is that we have increased amounts of precursors because they can't become aldosterone. So there will be some mineralocorticoid activity, not as much, but there will be some activity. So that's the whole idea here, is that we have intermediate steps. Okay, this is important that we have intermediate steps that exist, and these have some activities as well. So here's a nice table that will show us how you get from cholesterol to testosterone. Okay, and again, we have the steroid hormones, so they're made from cholesterol. So this is cholesterol, you can see, and then we have estradiol, cortisol. This is also steroid. So these are very important to remember. So you must remember cortisol, you must remember aldosterone, so testosterone, progesterone. How you remember this is you need to think about your adrenal cortex, okay? So if you think about your adrenal cortex, it's very easy to remember. And then you also have your ovary and your testis. So anything that is sexual. So sexual stuff and your adrenal cortex, okay? And your adrenal cortex is also producing androgens, by the way. And then the other one is your vitamin D stuff. And we're gonna see them later, okay? So here's another table of steroidogenesis. So we have cholesterol and we are making aldosterone here. And we're also making cortisol here. And we did already talk about the clinical implication. So yeah, that was that. And in this table, we here, for example, in this table, we have progesterone and androstenedione, okay? So here in this table, you can also see androstenedione, okay, here. So you have that here as well. So basically this is steroidogenesis. So basically vitamin D, as I said, is in its active form, it is calcitrol. And basically calcitrol will regulate the absorption of calcium and phosphate, okay? So that is a calcitrol. It's the active form of vitamin D. And it's also steroid. So the steroid hormones location, again, as I said, you need to think about your adrenal cortex and your ovary and your testis, and then you have your active form of vitamin D, the vitamin D cycle. Here we have skin, liver, and kidney. But we're not going to talk about vitamin D cycle too much. Now we're going to talk about amino acid derivative hormones. So we just have modification like hydroxylation are needed to make hormone from a single amino acid. So mainly we have tyrosine, okay? So you got to memorize tyrosine, but we also have tryptophan, but mainly we have tyrosine. So we have one amino acid, the tyrosine, and then we modify this and we get hormone. So here is a very simplified schematic, okay? So what do we have? Tyrosine, thyroid, okay? Very easy. You have tyrosine, thyroid. Then you have, from tyrosine, you get dopa, and then from dopa, you get catecholamine. So you get dopa, and then you make dopamine from that. But you also get norepinephrine and epinephrine. Then from tryptophan, you get serotonin, okay? So you get serotonin as well. And then from serotonin, you get melatonin, okay? Then from histidine, for example, you get histamine, okay? But the most important one that you need to remember here is going to be tyrosine and thyroid, and then tyrosine into dopa, okay? Also know tryptophan into serotonin, and yeah, once you know serotonin, melatonin is easy. And then the last one is a polyunsaturated fatty acid derivatives. So what is a polyunsaturated fatty acid? So this is just biochem recap. So you have saturated fatty acid, monounsaturated, polyunsaturated. So here in the saturated one, you do not have any double bonds between the carbon. But in a unsaturated fatty acid, you will have double bonds in the carbon here, okay? Because they're not saturated, okay? You can still add hydrogen to this, for example. If you have one, it's monounsaturated. If you have multiple double bonds, it is very, very, very unsaturated. So it's more than, so it's like polyunsaturated, okay? So here is a polyunsaturated fatty acid. You can see that we have multiple bonds. And these hormones derive from polyunsaturated fatty acid, and the most essential ones are your are going to be your thromboxane, prostaglandin, and leukotriene. Okay, so you're going to have arachidonic acid, and you have two pathways. You have the cyclooxygenase pathway and the LOX pathway. Okay, cyclooxygenase and a LOX. I don't remember LOX. It's lipoxygenase probably, but I'm not sure. Okay, anyways. So you get thromboxane and prostaglandin here. And then with LOX pathway, you get leukotriene. Okay, so we're not going to be talking about details of this, but basically this is a bronchoconstricting agent. This is involved in inflammation and pain, and thromboxane is very important for clotting. Okay. Now, hormone in blood. So how does hormone work in blood? Okay, so it could be either free, unbound, and it could be bound to a carrier protein. Okay, and basically these carrier proteins are made by the liver. Okay, so this is very, very important because again, we have a clinical correlate here. If you have a liver disease, if you have cirrhosis, you're gonna have problems. You're gonna have endocrine problems as well. Okay. Now, basically, the more you can dissolve yourself in water, the more you can be free and unbound. Okay. So imagine that you have an amino acid derivative over a very small peptide, right? So do you need something to carry it? Okay. The answer is no. You really don't need something to carry that because that is already going to be very, very soluble in your blood. Okay. So if you are a amino acid or a peptide, you are free and unbound. Whereas if you are lipid and big, also thyroid is peptide, but yeah, it's gonna be like amino acid derivative, right? From tyrosine to thyroid, we talk about that. So yeah, but it's still transported by specific glycoproteins. Okay, so you have albumin. Albumin can carry as well. Albumin is non-specific, but albumin carries hormones as well. Albumin is like the major carrier. It gives the blood its oncotic pressure, but also carries bilirubin, fatty acids, but some hormones. So the carrier proteins, you can have non-specific. So albumin does carry some hormones, around 10 to 20%. But the specific ones, we have, for example, sex hormone binding globulin, because sex hormones are lipid, steroids. So they can't dissolve in blood because blood is water, right? Corticosteroid binding globulin, again, corticosteroid, steroid. Then you have thyroxine binding globulin, TBG, CBG, and SHBG. And these are globulins that are produced by the liver. Okay, very, very important. Why do we need them? So basically, you need these carrier proteins because again, the first reason is you have water insoluble hormones that you need to carry. They also help you to reach your specific target. And then the last reason is because they act as a reservoir because they're going to be bound to it. So they act as a reservoir. Okay. So the hormone receptors. There are two types of hormone receptors. So basically, broadly speaking, you have your membrane receptor and your nuclear receptor. So imagine if you're a protein, right? If you are a protein, you will get bound to the membrane receptor. Whereas if you're a fat, you can transverse through the membrane because the membrane is lipid, right? So basically, you can get through the membrane and you can enter the nucleus, and then you will get bound to your nuclear receptor. So it's very, very easy, right? If you're water insoluble and lipid soluble, you go to your nuclear receptor. Whereas if you're water soluble, protein, peptide, you go to your membrane receptor. So the nuclear receptor superfamily, we have a nuclear receptor superfamily. You can see that we do have a bunch of these steroids. But we also have to have the thyroid. We're going to talk about that. But other than thyroid, we just have steroids, you can see. Even though thyroid is an amino acid derivative, it is special. So we're going to talk about that later. And we have the nuclear receptor superfamily, where we have a very conserved sequence. So you can see it here, okay? Just the molecular biology of it. You can see this. There's this concept, okay? Yeah, that's that. Now, the cell surface receptors is we have three types, right? We have the G protein coupled ones. So you can look at the slide and try to understand, you understand the pathways. Now, if you don't know these pathways, it doesn't really matter. because you don't need to know them all, okay? But if you want, you can do a recap on that. But I'm not going to explain every one, but I'm going to explain the important ones. Then you have your ligand-gated ion channels, and then you have your enzyme-linked receptors. So this could be a receptor tyrosine kinase or a receptor protein. So it could be like a serine kinase, so non-tyrosine, okay? So it could be receptor tyrosine or non-tyrosine for enzyme-linked. Then you have your G protein, which you have GS, GI, and GQ. And then you have your ligand-gated, okay? So the most important one is that you need to know is that the G-coupled pathway is very important. Mainly GS and GQ, and GI is basically the opposite of GS. But what GS does is that it activates adenylate cyclase, which converts ATP into cyclic AMP, which then activates protein kinase A, which will then go ahead and do various functions, okay? It can phosphorylate many targets. But basically, this is the main cell signaling pathway. For GQ, it will allow the conversion of phospholipase C, PIP2, because phospholipase C is the enzyme that is activated by GQ, PIP2 into IP3 and DAG. And IP3 will activate protein kinase C. And DAG will activate calcium, okay? And this calcium is bound to calmodulin, and then this calcium-calmodulin complex will activate many enzymes, okay? And basically, again, this will lead to an activity. So basically, all these pathways, either with pKa, pKc, or calcium calmodulin, you lead to an intracellular activity. And the net result, again, is we will phosphorylate stuff, we will activate enzymes, we will go to the nucleus and activate transcription factors and allow for gene expression, make proteins, so all that stuff, okay? And basically here, we have CAMK, which is calcium calmodulin dependent protein kinase signaling. Okay, so that's just the name. Don't get confused, but these are the net pathways of the G protein. Okay, so you can either get to pKa via the GS signaling pathway, or via the GQ you can get to PKC and CAMK. An example of this is the adenocorticotropic adenocorticotropic hormone. So basically we have the pituitary glands that will produce adenocorticotropic hormone. This goes to the adrenal cortex and causes cortisol production. And this is just dependent on the GS, okay? So the GS pathway is activated. We have cyclic AMP, we have the cyclic AMP production, and the cyclic AMP will activate pKa, and pKa is active, which will cause the events for the signaling of the cortisol, okay? So here we're going to see the molecular biology of pKa, if you want pKa anatomy. So it's a homotetramer, so it has 2 R subunits, which are regulatory subunits, and 2 catalytic subunits. And basically, the mechanism is cyclic AMP binds to this regulatory subunit, so cyclic AMP binds here, and this will create a change of conformation, releasing the catalytic subunit, which will produce the biological effect. Now, let's talk about role of endocrine system. So the role of endocrine system is multiple. It is regulatory. So it will regulate. What does it regulate? It regulates homeostasis, metabolism, growth, reproduction, and sexual function. Okay? Very, very easy. Now, which hormones do what? So this is important. So a little recap in hormones. So for body temperature, you have your thyroid hormones. For volume and ionic composition of body fluids, you have your antidiuretic hormone, aka arginine vasopressin. It retains water and vasoconstricts, so it increases your blood volume and blood pressure. Aldosterone retains sodium, does the same thing. It will increase blood pressure. Atrial natriuretic peptide will counteract the renin-angiotensin system, so it will do exactly the opposite of what renin-angiotensin system does. So yeah, here we don't have it here, but yeah, basically it vasodilates, it will push sodium out, so it will oppose antidiuretic hormone and aldosterone because it also pushes water out, and it will decrease the sympathetic tone. Because the renin-angiotensin system, angiotensin 2, does the exact opposite, okay? Then we have parathyroid hormone, which will increase the calcium in blood. And basically, this is very, very essential. How does it do it? It will stimulate osteoclast and break bone to get calcium in the blood. It will stimulate the conversion of vitamin D to its active form calcitrol. And what does calcitrol do? Calcitrol increases intestinal absorption of calcium and phosphate. But parathyroid hormone will also promote phosphate excretion in kidney because phosphate in blood binds to calcium, which will reduce its availability. So we don't want this happening. Now, calcitrol, we said it will increase the calcium and phosphate absorption in the intestine, and it's essential for bone formation because you need hydroxyapatite, which is made up of calcium and phosphate. And calcitrol also has a negative feedback on the parathyroid hormones. So you have a negative feedback because it stimulates calcitrol. So you have a negative feedback going like this, okay? So here's the vitamin D cycle here. We see that we have cholesterol, and then basically we have cholecalciferol. So here this will be converted in the liver to 25 hydroxyvitamin D3, and in the kidney it will become calcitrol, okay? Now, the arterial blood pressure and cardiac activity, we have monoamines, and monoamines are categorized in two categories. We have catecholamines, which are norepinephrine, epinephrine, dopamine, and indoleamines, which are serotonin and melatonin is made from serotonin. So these are made from tryptophan, these are made from tyrosine, remember that. So here we have epinephrine, norepinephrine, vasoconstriction, right? And dopamine at low dose, it will vasodilate, and it will at high dose, it will vasoconstrict. And it at very high pharmacological dose, it will increase your cardiac output and basically cause your heart to pump, okay? And basically, it will also cause renal artery vasodilation, dopamine. Now, angiotensin 2, potent vasoconstriction, stimulate antidiuretic hormone, aldosterone, and also endothelin. It will cause cardiac and renal remodeling, which is a completely complicated topic. I mean, it's not that complicated, but it's another topic, so we're not going to talk about that in this lecture. And it will increase thirst and salt appetite. So the idea of angiotensin 2 is that it will increase your blood pressure and your blood volume a lot, okay? It's very potent, and it will increase your blood pressure. Then you have endothelin, which is produced by endothelial cell. It's a very potent vasoconstrictor. Then you have your atrial natriuretic peptide, which is AMP, and this will counteract your RAS. We talked about this before. Then you have your prostaglandins. So your prostaglandin, you have your PGD, PGE, PGF, and PGI, and also your thromboxanes. So your thromboxane will vasoconstrict, but your other prostaglandins, mainly the E and I, will cause vasodilation. And basically, yeah, so you can remember this as DEFI. So DEFI, okay? Just a little mnemonic. Now your stress response. So stress response is your adrenocorticotropic hormone, so ACTH, and this will cause cortisol release, and cortisol will reabsorb sodium, okay? Very, very important. Just like aldosterone, you reabsorb sodium. And then you have your promoting gluconeogenesis in liver to increase your blood glucose because you're in a stress state, okay? It will also be anti-inflammatory, immunosuppressive. Why? Because all your energy should go to your muscle and brain because you're in a stress situation. In a stress situation, you don't want your immune system wasting energy, okay? You want everything in your muscle and your brain, okay? And will also stimulate norepinephrine release, obviously, right? Because you're in a stress situation, you gotta fight. All these mechanisms are designed from an evolutionary perspective, okay? So you gotta fight a lion, you gotta run away, you gotta hunt. So all that stuff, that's a stress response. So you need the energy in your brain, you need the energy in your glucose in your muscle, okay? You don't wanna waste energy. That's why it's immunosuppressive. You don't want white blood cells energy. You don't want the white blood cells to waste the energy that you need to give to your brain and muscles. Then you have your epinephrine, and epinephrine is mainly a hormone, but also a neurotransmitter, obviously. And basically, it is allowing for vasoconstriction, vasodilate the heart and the vessels that go to the skeletal muscle, obviously. It will also cause bronchial dilation because you want to have maximum air, maximum oxygen, right? It will promote breakdown of glycogen, so you want to increase your blood glucose. So the whole idea about stress response is increasing blood glucose, increasing the blood that is going to your muscles and your heart and your air that's going to your lungs, all that stuff, okay? Norepinephrine also causes bronchodilation, so it's the same idea, okay? Then for metabolic regulation, you have gastrin, which is produced by G cells, so this will stimulate the secretion of gastric acid, so you gotta help yourself digest. Cholecystokinin, eye cells in the duodenum stimulate pancreas to release its enzymes and gallbladder for bile. So you get that bile, got that enzyme, so you can digest everything, okay? The last one is secretin, and it will protect the duodenum from acid. So it will inhibit gastric acid secretion, and it will stimulate the pancreas for more bicarbonate, okay? It kind of says I want more bicarbonate because I gotta protect myself from that acid that's incoming. The next one is growth and development. So you have your growth hormone, which is produced by the pituitary. You have your thyroid hormone, which is also involved in growth and also in neuropsychic development. This is very important. You have insulin-like growth factor 1. This is also important in growth of bone and muscle. It has increased glucose uptake, neuropsychic development. Steroid hormones are essential for growth like testosterone and sexual development. And also parathyroid and calcitrol are important here. This stimulates this. This has a negative feedback on this. And this will cause bone growth, okay? So again, bone growth is growth, right? So that's that. Sexual functions, you need testosterone, like for gametogenesis, you need a gonadotropin. Oxytocin, you need it for delivering the baby and also bonding with your partner. So those are sexual functions. Now, how do we detect hormones? What are the lab methods? So the first method is the immunoassay method, and the idea here, the logic here, is using antibodies to bind to a hormone. But it binds to an indirect feature of the molecule, okay? You are binding to the antigenicity, okay? You are binding to the antigen. So the feature here is antigenicity. So it's an indirect feature of the molecule, okay? So the first one is radioimmunoassay, where you are using a tracer antibody with a radioactive molecule. And here's the antigen, so here's the hormone. You have a capture antibody and a tracer antibody. And this is basically non-competitive. You measure the radiation and you understand the hormone, okay? Whereas in competitive, you have a synthetic hormone with a radioactive tracer and the patient's hormone. And they compete for the antibody, okay? And then you wash this sample and measure the radioactivity, okay? If you have a lot of radioactivity here, this means that the patient doesn't have the hormone because it couldn't compete, right? All of the antibody is occupied by the radioactive synthetic antigen. Whereas if you have low radioactivity, it's the exact opposite. Whereas it means that the patient has a lot of the hormone and it actually competed with the synthetic one, and it allowed for it means that you can actually quantify it by measuring the radioactivity. So this is really, really good, okay? This is radioassay. Then you have ELISA, so enzyme-linked immunosorbent assay, where instead of a radioactive tracer, you have so I'm going to do the direct, okay? So you have your antigen, and then you have your antibody, which is linked to an enzyme which cleaves the substrate, and this then you can measure because the enzyme, it will cleave the substrate into a new product, okay? So you can then measure the amount of product, which will allow you to measure the amount of hormone, okay? It's the same thing. And then you also have like sandwich ELISA, where you have like a capture antibody, the antigen, another antibody, and basically another antibody that is linked to this. Or you can have an indirect assay, where you have your antigen, primary antibody, and secondary antibody conjugate, okay? So that's that. And here, as I said, it's a sandwich. So you have a capture antibody for the antigen, you have another, and then you have like an indirect one, okay? You have a primary antibody and a secondary one that's linked to enzyme, but it's the same idea. So basically, the problem here is specificity, okay? It's a problem. The specificity, it's not that high. So these are the immunometric assays. You can see some examples, but they are not specific. They are not specific because you are looking for an indirect feature of a molecule. So it can underestimate. For example, you can have matrix interference, which is when an other molecule interferes with the binding of the antigen. Or you can have an overestimation, whereas very structurally, molecularly similar molecules are binding to the antibody, causing you to detect more than the actual amount. Or you can have an antibody in the patient's blood binding to the detection antibody, causing interference, and this is called heterophilic interference, okay? So again, it's not specific. There are problems. How do we overcome the problems? We do liquid chromatography mass spectrometry, which measures molecules according to their mass and charge, okay? And ionize the samples with electron beam. We're not going to go into detail. You don't need to know the detail, but you just need to see this graph here, where you have You're identifying molecules with very nice specificity, okay? So here, again, we have a comparison. Here's a testosterone measurement. So in males, you got a lot of testosterone. So the specificity is okay, right? So here we have the real values with mass spectrometry, and then we have the value with the immunoassay, and they're both correlating. But here there's no correlation because in females testosterone is low, so you need much more specificity. And basically mass spectrometry has the specificity, okay? Whereas the immunoassay doesn't. So we use mass spectrometry, that's the gold standard. Now, the last topic before we finish this lecture is secretion rhythms. And basically, we need to do some definitions here. The biological clock is your physiological mechanism that determines your rhythmicity. So this is like your thirst, your food seeking, your sleep-wake cycle. Okay, this will determine some sort of rhythmicity. And your biological rhythm is your cyclic and repeated variation of that physiological function. Okay, now we have different rhythms. Okay, so we have circadian, which comes from the Latin circa, which means around, and dian, which means day. So this means it's just one day. Okay, so circadian is 24 hours, whereas ultradian, okay, so think about ultra. So it's much faster than 24 hours, okay? It's basically faster than 24 hours, whereas infradian is infra, so it's below a day, okay? So it's longer than a day, okay? It might sound kind of paradoxic, but if you think about like this, it's very easy to memorize, whereas like infradian is much slower, okay? So infra in sense of like being slow, so it takes longer. So yeah. And why do we have circadian clocks? We're going to talk about this later, but basically they allow for the fitness and survival of the species. So again, whenever you're thinking about your physiology, you need to always come from an evolutionary standpoint, and that explains a lot of stuff, okay? So you need to survive, and we're going to talk about how this works later. But here are some examples. So here's an ultradian rhythm, EEGs, your brain, your cardiac activity has a rhythm, your respiratory rhythm has a rhythm, and they're very, very fast. Your circadian rhythm is your sleep-wake cycle, obviously, and then your infradian rhythm is your menstruation, so it's a 28-day cycle for a woman, and hibernation, for example, is for bears, for example, it's very, very long. Now, here's an experiment. So this experiment, basically, I'm going to explain. So there's a plant, okay, and the plant is in front of a window, and it sees the light in the day and dark in the night. And basically the behavior of this plant is that the plant has its leaves open in the day and its leaves closed in the night. So what happens then is that the experimenter will block the light, okay? So now it's permanently dark. So there's a stable condition. The outside condition doesn't change. It's stable, it's always dark. But what happens is that the plant still opens its leaves and closes its leaves according to time, okay? So before it was like 12-hour light, 12-hour dark, right? It opens 12 hours, close, and then the last 12 hours. So it had like a circadian rhythm that was influenced by the external environment, but now even though the external environment doesn't provide the necessary stimulus, it still relies on its inner biological clock to produce its own circadian rhythm. So this is the idea of the circadian rhythm, okay? So very, very important. Now, you have the suprachiasmatic nucleus of the hypothalamus, which is the main biological clock in the humans. And this is the pacemaker. It is the central pacemaker. Now, you obviously have a bunch of peripheral pacemakers, which we're going to talk about. For example, your heart, your sinoatrial node is a peripheral pacemaker. But your central main pacemaker, your main biological clock, is your suprachiasmatic nucleus of the hypothalamus. And the ganglion cells in your retina will give info to the suprachiasmatic nucleus through the retinal hypothalamic tract. Very easy to understand why. And basically, this will signal light, okay? So here's a graph that we can see. And here is, for example, light that comes to the suprachiasmatic nucleus, and you don't need to know this detailed, but basically this light info will inhibit the melatonin, okay? So whenever there's light, you don't produce melatonin. Whenever there's dark, you do produce melatonin, okay? So this is a graph. So here you have your suprachiasmatic nucleus, and it will signal everything, okay? Everything. And here's the big question: why the heck do I need an inner clock? So that was the question I asked before, and the answer is simple, okay? So there are three reasons. First, reaction to stable conditions, okay? So basically, if you are living in Norway, let's say you are in Norway, okay? And basically Norway, in the summer, you're going to have sunlight for 24 hours, okay? Now, if you don't have a biological clock, you will not be able to sleep in Norway. But because you have a biological clock, you get sleepy, okay? At the same time that your biological clock is used to. So this allows you to react to stable conditions, okay? Stable environmental outside conditions. It allows you to anticipate predictable change. So I don't wait to get food until I get hungry, okay? I anticipate that I will be getting hungry at 7 p.m. because that's how I eat. That's when I eat dinner every single day, for example. So I eat at 7 p.m., okay? And the last is coordination and synchrony in the entire human body, in your entire physiology. And here you can see that all the organs are affected by the central pacemaker. And this is very, very important. So it's very, very important. So your inner harmony, okay? So the molecular circuitry is very easy. You have your clock gene and your BMAL1 gene, and they make clock protein and BMAL1 protein. And basically, these clock and BMAL proteins will heterodimerize. So they're just going to dimerize, they're going to be together. And then they're going to sit on the E-box region of your DNA. And this will act as a transcription factor. And this transcription factor will produce needed proteins, but also two other proteins that are per protein, which it comes from period, and your cry protein, okay? And then these will dimerize and inhibit the clock protein, okay? And the BMAL protein. So it's very, very easy, very simple. And this creates rhythmicity. So I'm going to show you how this creates rhythmicity. So here we have a nice graph. So on the y-axis here, I have the per cry concentration in the cytoplasm of the cell, okay? So right here, we have the clock and BMAL protein starting to transcribe per and cry, okay? So at this point, per and cry is being transcribed, okay? And then its concentration in the cytoplasm increases. And at the peak, it starts inhibiting this complex. So then the transcription process is halted. So then its concentration goes down. And at some point, it goes to the baseline. And at this point, what's going to happen is we will have, again, the activation of this system. Because now this system, it doesn't need to be repressed anymore. So then this system is back activated. And it's the exact same thing that repeats. So here you have a clock cycle. So you can see the rhythmicity, okay? This creates rhythmicity, okay? So here's rhythmicity. So let's say this is the beginning, this is the baseline, and you can think of it as a clock, okay? So basically you can think of it half of the clock. So like this, okay? So it's exactly like a clock. There is a rhythm. Very, very easy. This is the molecular circuitry, okay? Very, very easy, simplified. You have your clock and BMAL, which produce per and cry, and then these will go ahead back to inhibit it. And this creates that rhythmic oscillations, okay? Now, rhythms in physiology. This is very important. So several systems in the body have their own circadian rhythm that act in the window opened by the central circadian rhythm. Examples, livers, HMG-CoA is maximum expressed at night. So statin, which is the inhibitor of this, you gotta give it at night, right? Because to get maximum efficiency. Early morning, you got cortisol release. Your blood pressure at early morning is high because why? You have incoming cholesterol. Incoming cortisol and incoming catecholamines, norepinephrine. Why? Because your body tells you to get ready to fight. Okay. In the early morning, you have your stress response that is increasing because you have a day. Okay. You need to hunt. You need to get food. You need to survive that day. Okay. That's the idea. Then you have your peripheral molecular clock, and this is very important. So 10% of the entire genome is under clock control. Okay. Very, very important. Entire genome, 10%. 50% of the entire protein coding genes have circadian oscillations. So 10% of the entire genome, which is made up of lots of non-coding DNA as well, but the 50% of the entire protein coding genes have a circadian control. And also the post-transcriptional and translational processes are also under circadian clock control. Okay, so that is the peripheral molecular clock. Now here's an example of melatonin. It gets peak during night at darkness. Okay, it's very low at early morning. Early morning, and it starts producing at the evening, and then, you know, after you sleep, it goes down. Okay. Some examples of cortisol. So it starts increasing at night, and it peaks at 4 to 5 p.m. Okay. So when the patient wakes up as well. So the idea here is that you want to have that stress response when you wake up. When you wake up, you want to be fresh. You want to have that norepinephrine going. You want to have that heart. You know, you want to have that air in your lung, because you're gonna start your day. You're gonna go get breakfast. You know, all that jazz. Okay. And here is growth hormone. So here we see peaks in the male and the female. So again, we have time. So yeah, that's peaks in the male and the female. So yeah, we see that it is going to be peaking here when you're sleeping, for example. And testosterone also peaks in the early morning as well. So it is very important because, again, testosterone has two functions, the sexual function and the anabolic function. So again, you're waking up, you want to have that energy, okay? You want to have that sexual seeking behavior. You want to get that, you want to reproduce. That is very, very important for biology, for evolution. And then you want to prep yourself for the hunt. You want to prep yourself for the survival. You want to prep yourself for the fight, for the run. So your stress response, your sexual testosterone, which is not only sexual but anabolic, you want to get that in the morning as well. So again, testosterone peaks in the morning, cortisol peaks in the morning, melatonin peaks in the night, in darkness, and growth hormone peaks as well in the night. So that is it for our first lecture. So I'm concluding the first lecture. I hope you guys enjoyed. If you guys liked, you can leave comments down below. You can interact. And I'll see you on the second lecture of endocrinology. See you.